The overall goal of this methodology is to acquire and analyze the dynamics of a large population of adherent cells monitored by means of lens-free video microscopy.
So we think that this technique is interesting in many fields of biology, for studying cell proliferation or cell size studies, or drug screening, or different bioprocess monitoring. So this technique is label-free. It's very simple, but it's very effective in studying large cell populations.
We had the idea for this method when we shifted from the field of detecting bacteria to the field of imaging mammalian cells. Then it was obvious that the biologists, they will need time-lapse acquisition of the cell culture directly into the incubator. Then we built a customized lens-free video microscope that is compatible with the incubator, and we developed an holographic reconstruction algorithm to get the picture of thousands of cells that are present in the very large field of view.
Begin this procedure by coating a 6-well glass bottom plate with fibronectin at room temperature for 1 hour. After 1 hour, remove excess fibronectin and rinse out the 6-well glass bottom plate with PBS. HeLa cells, grown in DMEM plus glutamine medium supplemented with 10% heat-inactivated fetal calf serum and 1% penicillin and streptomycin are used in this demonstration. Seed 20,000 cells per culture plate. It is important to use a container with a glass.